Well, thank you, Sunny, and thank you all for coming out on this damp evening. I feel very much at home with the cold air temperatures and the fog and the dampness. It's just about right. I feel like I'm back at the time. As Sunny mentioned, I've been involved very much in the Census of Marine Life program, particularly the last 4 years of that program. And for those of us who were involved, it was really the opportunity of a lifetime. And so I'm going to tell you what I can in about 45 minutes, which is just, again, scratching the surface. But I hope it illustrates that it was really an amazing program, produced some amazing findings, and also opens up some new opportunities. One of the other reasons why you should attend lectures in person is that speakers like me wander around as we talk, and so when you're trying to film it, we drive people crazy. Now, they put me on a short leash, so I know what my dog feels like, but I will hopefully not trip over it as I give the talk. Now, I think this audience is particularly aware of the fact that we should probably be calling planet Earth planet ocean. Something between 95 and 99% of the habitable habitat on Earth is in the oceans, and something like 70% of the ocean's surface is covered by ocean environments. So Arthur C. Clarke, always very prescient in his writings, had it right when he said how inappropriate to call this planet Earth when it is quite clearly ocean. And so this is a cartoon, but I wanted to put it up to illustrate what most of the ocean looks like. And so if you're going down in a submersible, as I've had the opportunity to do in a few instances, you may luckily capture something like this, but mostly it's complete darkness. There is no sunlight penetrating through most of the ocean depths, and the abundance of organisms is very scattered. And eventually you reach the seafloor and see this sort of habitat. So what you see here is the most abundant habitat on Earth by far. And that's what it looks like. It doesn't look like much, but in fact there is a huge diversity of species that live between these grains of sediments, and that's what really gets me personally very excited about the ocean. But there are other people who deal with other aspects of ocean life. And so this is, in fact, what the Census of Marine Life faced. And so if you followed any of the press associated with the program, you've seen the emphasis on new species. And there were lots and lots of new species, some of them very beautiful and spectacular, as you can see in the images that are popping up here. But that wasn't all the census was about. But I wanted to introduce it because I know that's what a lot of you heard about. And some of these things are large, like kelps. And in total, there were something like 4 to 5 new species of marine life described every day. And so the limitation there is not in terms of the diversity of life, but in the experts who are available to describe this new life. That's really the rate limiting step. And so with 4 to 5 new species every day, that number has actually gone up during the census. There is clearly lots and lots to do. And so that's the starting point for the census. Lots of diversity, lots of unknowns. And so what do we do about it? Well, I think what I would like to say, and there's some students in the audience, is that I think it's a great time to study ocean life. And there are 3 reasons for this. And these are all good news stories. There'll be some bad news in just a moment, but we'll start off with the good news. First of all, there are lots of great new tools. In the last decade, and a little bit beyond that, we've developed some really remarkable means of sampling the ocean. And so our capacity to understand life in the ocean is really much better than it was even a decade ago. There are lots of new discoveries, some of which you saw on the previous slide, that I think have created a lot of excitement, increased awareness among the general public. And this, I think, will create new opportunities because people care about things when they see charismatic species in the ocean. And so there are lots and lots of them, and I'm hoping that's going to help carry us forward. Now, there is lots of bad news, and I'm not going to dwell on this because we could spend many hours doing so. But this slide that you see in the inset shows you the changes in atmospheric carbon dioxide measured in Hawaii since 1960 through the present. And you can see it's gone up very substantially. Importantly for life in the oceans, this has also caused the CO2 in the oceans to go up, which in turn has caused the acidity to decrease. So the oceans are becoming more acid, they're becoming more warm. We can add to this fishing effects, pollution, nutrient-loading disease, invasive species, loss of habitat, and changes in hydrology as fresh water comes into the coastal zone. So all of these things create some real concerns about sustainability of ocean life and how the oceans are going to persist into the future. Some of you may be aware of a very intriguing paper published by Ben Halpern in Science that showed that the cumulative effects of human impacts are actually much more substantial than the summation of the individual components. So if there are many impacts upon you, they're not simply additive, they actually create a bigger effect than you predict through simple addition. Now, being a Newfoundlander, I have to tell a classic story of codfish. And so this is a little boy about 1914 in Battle Harbor, Newfoundland, And you can see this boy is standing next to these really impressive codfish. We're near Cape Cod, and so I think I have a very sympathetic audience. Now, when I was a boy of about this size, I used to occasionally go out fishing with my grandfather, and we would catch fish that were about half the size of these humongous fish. And I thought that was the norm. That was what, to me, seemed like an appropriate baseline for what the oceans ought to look like. It didn't occur to me that, in fact, the fish used to be much bigger. Now, if you go out there today and catch fish, I'm a little older than I was at this age, you'll see smaller fish still, probably half the size of what I saw as a child. And so the point here is that what we think of as the norm, so for example, when John Cabot sailed across the ocean and came into Newfoundland waters, threw a basket over the side and it came up filled with fish, that was the norm at that time. But clearly now we need huge trawlers, they rake across the seafloor and finally do catch many fish, although in the case of Newfoundland we saw a complete collapse of the cod fishery in 1992, and what that led to was 20,000 people lost their jobs overnight. And that was in a population of about 500,000 people. So it was the largest industrial disaster in Canadian history, and it was driven essentially by the loss of a single marine species that had collapsed due to inappropriate management and over-harvesting. So that's changing baseline. So the ocean, as it looks today, is in fact very different than the way it used to look before. And though many of us tend to think of it the last 50 years, that's not the way to think of it. As you'll see later on in the talk, some of these impacts go back many thousands of years. One other little anecdote I wanted to tell you, as long as we're talking about Newfoundland, this is a walrus. And I attended a talk a number of years ago by Ransom Myers, a very influential fisheries biologist who died quite young of cancer. But Ram started talking about the role of walrus in Newfoundland waters. Now, I had grown up in Newfoundland. I had been studying Newfoundland ecosystems for about 15 years, and I did not know that walrus had once been part of that ecosystem. So once again, these changing baselines where we're biased by our views today of what the oceans ought to look like. So the walrus are now gone, they're not part of the system, and most people don't even know that they were ever part of the system. So along comes the Census of Marine Life. And so the census was about coordination, cooperation, and collaboration. There were a total of 17 different projects organized around the world. 14 of these projects were oriented in field programs. 3 of them were more integrated in terms of the work that they did. So there were 5 in the deep sea. We had projects working all the way from the intertidal, including the Nagisa program that Sunny mentioned at the onset, all the way down to the abyssal plains and even into the trenches. We had researchers working on microbes all the way up to whales and working all the way from the tropics to the poles. And so in total, these 17 projects create this sort of footprint around the global ocean that really is impressive in terms of the scope of the research. And this all started relatively modestly in 1996. Fred Grassley, a deep sea ecologist, somebody I've worked with quite a bit, walked into the office in Woods Hole, not too far from here on the Cape, and said, we've got a problem. There's a lot of marine biodiversity. It's changing, and nobody seems to care. What can we do about it? So he and Jesse chatted, and they worked on this, and within a very short period of time, they had come up with the Census of Marine Life. And the initial idea was, again, as Sunny mentioned, to count all the fish in the sea. But Fred actually cares mostly about invertebrates. And so the program quickly expanded and really became all forms of marine life. And so counting and enumerating everything out there. Now, it was actually also a very interesting model for doing science, because the way it worked is that the Alfred P. Sloan Foundation in New York City invested something like $70 million in the census. And the majority of that funding went towards bringing scientists together to agree on questions and methods. And those scientists would then return to their home countries. So they're brought in from all around the world. In total, there were more than 80 countries and more than 2,700 scientists involved in the census. So Sloan would bring these people together, they would agree on the questions and the methods, and then they would go home and raise support. And in the end, there was about $650 million spent on the Census of Marine Life. Now, this includes ship time, which is very expensive. You can easily spend $50,000 a day on an oceanographic vessel. But the key thing is that it created a truly global effort. And so it meant the scientists were sharing methodologies, and they were creating data sets that were truly integrative. And so they used the same techniques. And so if you got 2 different numbers from different areas around the world, you could directly compare those numbers without worrying whether it was an artifact of the particular approach. So the census revolved around the changing oceans, and particularly oceans past, present, and future. So oceans past, as I've already alluded to, there's tremendous productivity of the oceans that we've lost. What you see here on the left-hand side are some gentlemen standing in a shell pile of abalone in California. This is actually somewhere near Santa Barbara. Now, you may know that abalone is now an endangered species, and yet look at the abundance of the abalone shell that these guys are shucking. On the right-hand side, this is actually a market in Denmark. And you can see all of these tuna that are laid out on the floor, very large fish, very abundant. You'd be hard-pressed now to catch tuna in the North Sea, and yet at one time they were this abundant that they could create this tremendous market in Denmark. The ocean's present is actually a very complicated story because it's a story of both the good and the bad, and you'll see some of that throughout the talk. The good news is that there's still tremendous beauty and diversity remaining in the oceans. And so we can go to some habitats and we can see very productive areas, very diverse areas. But we can also see this sort of result. And what this is, and this may make you gasp, this is actually a deep sea trawl from the Mediterranean which collected more garbage than it did animals. Now this is the Mediterranean deep sea. The deep sea is what we consider to be among the most pristine ocean environment left on the planet. And yet you see this sort of catch. Now, clearly this is an extreme example, but certainly in the dives I've done over the years in areas from New England all the way up to Newfoundland and down to the Caribbean, it is rare to go on a submersible dive and not to see garbage somewhere during the dive, even in deep areas of the ocean. And so that's very sobering. So ocean's present is really a mixed bag. Ocean's future, we don't know, but one thing that we can say for sure is ocean usage is very likely to increase. And so we're going to put more pressure on the oceans, and the question is, what will those oceans look like as a result? So we'll say a little bit more about that. So most of the census projects focused on the oceans present, trying to sample different groups of organisms in different environments, but there was a program called the History of Marine Animal Populations to try to look back in time, and there's another program called the Future of Marine Animal Populations that projected forward and tried to look at what the oceans will look like in the future. Now, one of the big issues in terms of sampling the oceans is changing technology. And some of you may be very familiar with this sort of image. It shows you the bathymetry of the seafloor. And you can see all these tremendous structures, this great detail that we now know exists on the seafloor. And the inset here shows you a satellite image of phytoplankton productivity in the North Atlantic. So here's Cape Cod over here. And you can see this very detailed structure, very intriguing. And this sort of image tells us a lot about the oceans. So today we know this. We know what the seafloor looks like in a general sense, all this topography. We know what these circulation cells look like and the productivity of the oceans. But if we go back even just a little over 100 years to the Challenger expedition, this was the first really big oceanographic expedition, 1872 through 1876, this would have been completely unknowable. They had no idea that we would someday be able to create these very, very detailed maps of the seafloor and also to have satellites catching essentially images of thousands of square kilometers of ocean in very short periods of time. So at the time of the Challenger expedition, this sort of information was simply unknowable. There's no way we could conceive of even knowing it. And so through time, through the last 100 years and more, we've moved a lot of what was once unknowable into what is either unknown or known. And I bring this up because there are still things about the ocean that are unknowable. And so I would say one thing that is unknowable in any of our lifetimes, including the youngest people in the audience, is exactly how many species there are on the sea. That is unknowable. The reason for that is because the diversity is so huge and the habitat so complex, we are not going to be able to sample it all, no matter how much effort we try to put into it. We will get better and better estimates, and I'll come back to that issue a little bit later on. But the point is they're still unknowables. And so the key, I think, is to focus on moving things from the unknown into the realm of the known. And that's really where the census has tried to work. So taking on solvable problems in essence. So satellite oceanography has really revolutionized the field. And so we have satellites that look down on the ocean, produce these spectacular maps. Many of you have seen these, I would imagine. This shows ocean primary productivity. You can see the North Atlantic very much more productive than the tropical Atlantic to the south. But the problem with satellites is they see quite widely, but they do not see deeply. They tell us a lot about the surface, very thin layer of the ocean, but very little about the ocean beneath it. And so for all the other forms of ocean life, other than phytoplankton, the plant-like cells that drive the food chain in the oceans, satellites don't help us out very much. They tell us a lot about physical oceanography, they tell us a lot about phytoplankton, but that's about as far as it goes. In fact, we think of water as being relatively clear. The truth is, water is actually quite opaque in many ways. If any of you are scuba divers, you know that even in the clearest water, you'd be lucky to be able to see 100 meters. And so light dissipates actually very quickly in the ocean, which is why most ocean habitat is pitch black.
Could you comment on why there are certain areas, hotspots, shall we call them?
Yes. So the ocean is very uneven in its productivity, and the reason for this is the amount of nutrients that are available to the plants. And so just as you would fertilize your lawn or your garden, there's some areas of the ocean that naturally have higher nutrient levels than other areas. And so this is because of a variety of oceanographic features that we won't get into the detail of. But when you get to the extremes, toward the poles, that's where light becomes limited, because of course you have 6 months of relative darkness and then 6 months of light. So those waters tend to be relatively nutrient rich, and when you transport those waters towards the equator, they bring nutrients with them. Keeping those nutrients in fresh supply is what creates ocean productivity. And if that ever shuts down, the oceans will collapse. And so we have these areas that are much more productive than adjacent regions for that reason. Really, nutrients are the main driver. So we've used a whole variety of techniques over the years to try and sample what was a once opaque ocean. And so things like plankton nets, which we tow on the surface waters and deeper layers to collect zooplankton, have been around for 100 years. We use water bottles to collect nutrient samples and also microbes and phytoplankton. And then we use various coring devices to sample the seafloor. Again, many of these technologies are not that new. Multicore has been around since the 1960s, and these other techniques, as I say, are even older. But in the last 10 to 30 or 40 years, we've greatly expanded the range of tools that are available to us to try and sample the oceans. So submersibles, as you can see here, these have been used by scientists since the 1970s, and they are a critical part of our toolbox, particularly in the deep ocean. But today we take it even further. We have remotely operated vehicles called ROVs, which are essentially robots that we can send down to the seafloor. They're attached to a tether to the surface ship, and we can actually sit on the surface ship and watch it as it moves around the seafloor. A couple of years ago, there was a cruise on which I could not participate because I had a conflict in scheduling. And thanks to satellite technology, I was able to sit in my study at home with my dog curled up at my feet and a cup of tea in my hands, and tell the pilot exactly where I wanted the sample collected. And so he was doing that 500 miles away off the coast of Newfoundland while I was sitting in my bathrobe telling him what I wanted done. And that's the sort of technology that we have available to us today. We also have autonomous vehicles, and these are robotic vehicles. We program them, send them out on a mission, they collect their data and come back and tell us what they've collected. We have various acoustic devices that send out sound waves that tell us what bottom currents look like, and actually the currents all up at different layers in the water column. We have scuba divers, of course. That's been a technology for a very long time. And then we have these listening curtains that you see here. And I'll talk a fair bit about these a little later on because they tell us a lot about the movements of fishes. So this arsenal of tools is so much greater than it was even 10 years ago. Some of these tools were developed by the census, others were simply adapted by the census, and others have been around a little longer. Now, I mentioned these 4 to 5 new species every day. And so that translates to about 1,650 new species in the oceans every single year. And if you look at the bias, what we see here, these top 3 groups, the crustaceans and the mollusks, so things like shrimp and crabs are crustaceans. Mollusks include the bivalves, scallops, those sorts of things. And then the Pisces are the fish. Now, what you might try and infer from this is that these are probably the most diverse groups. This is actually not true. So first of all, there's a lot more effort put into trying to describe new species of fish than there are in other groups of organisms. People just get more excited about big animals that they can see. The second thing is that in the mollusks and the crustaceans, there is a very vibrant amateur taxonomy community. And these are individuals who are not professional scientists, but they enjoy doing amateur taxonomy, and so they go out and describe new species. And they're a very important part of why these 2 groups are among the most numerous in terms of new species. Now I wanted to point out to you here the bacteria and the archaea, and also the nematoda. And these are groups that don't look nearly as important in terms of their diversity, but in fact they are hugely diverse. With the microbes, and this is a point, again, that Sunny alluded to, and I'm going to come back to it, we now realize that the microbes are hugely diverse. And so the limit here, again, is a taxonomic effort, not the diversity that's out there. And we're just starting to appreciate it for reasons I'll show you in a minute. The nematodes are these little threadworms. And so these little threadworms, again, something you may think you don't care about, but they can be economically hugely important in agriculture. They're extremely species rich. Some of them are parasitic. So there's something you don't hear a lot about, but they do have important roles. And here you see these are relatively far down the list, but as I'll show you later on, very much underdescribed. So many of these groups were just starting to describe them and scratch the surface. So from hydrothermal vents alone, as you can see right here, every 2 weeks there's a new species described for hydrothermal vents. Now this is a very specialized environment, very small proportion of the Earth's surface that these environments cover, and yet we can still produce new species every 2 weeks. Again, limited by the taxonomists, not by the availability of specimens and new diversity. A couple of little quick anecdotes, I could give you pages and pages of these, but I just want to give you a couple to illustrate a few points. First of all, there's a study done in the Southern Ocean, so down near the Antarctic, where they found that 90% of the isopods, these are a type of crustacean, small crustacean, 90% of them were new to science. So 9 out of the 10 specimens that they looked at were new to science. This added 6% to the global total of known species for this group of organisms. This is just from a couple of cruises within a very short time period. So the potential, again, for diversity is huge. 106 microbial species in a single drop of seawater. This is the point we discussed earlier. I put species here in quotation marks because species for microbes is a little bit of a loosey-goosey concept, because where you define species in the microbial world is a little different than you do in the animal world. And so we typically say more like 160 microbial forms or types or something like that. And then there's a really interesting study. This is actually a scientist, Nancy Knowlton, who works at the Smithsonian in Washington. And she sampled coral heads, small coral heads, from an area of about 30,000 square kilometers, but very small chunks of coral from this large area in the tropical Pacific, in an area called the Line Islands, which are very poorly known. And she found more crab species in what amounted to an area the size of that desk, or a little bit larger than that, than in all of the Mediterranean Sea. And so we've been studying the Mediterranean for a very long time. It's a much bigger environment. It's actually 30 times larger than the area she was sampling. And yet with that small sampling effort, she was still able to find more species. So it illustrates just how much diversity is out there and how much is left to be described. So this may raise the question for you, how many species are there in the oceans? I'm going to tell you we don't have the answer. I can give you an estimate, but I will tell you how one goes about trying to come to some sort of estimate for that. So one of the things that we use to try and figure out how many species there are is something called a discovery curve. And so what we look at is the rate of discovery of new species as a function of time. Now, if we go out and we collect specimens, and we go out, let's say, in Boston Harbor, and over many years we keep collecting specimens, eventually we will reach a point where we stop collecting new species. We'll find more specimens, but we know everything. So we know all the diversity, and so that line will level off. So that is either a taxonomic group or location where we know all of its diversity. So that's fine and dandy. The problem, though, is if the curve doesn't start to level out, what do you do? So if you look at this particular curve, we could say, well, maybe it'll level off near the top of the screen, maybe it'll level off up here somewhere, or maybe it'll shoot through the roof of the building before it starts to level off. We don't know. And there is absolutely no statistical means of trying to figure that out. Now, to put this into practice, we'll start with the fishes. This is a relatively well-known group. Some of the fishes we've known and harvested for a very long period of time. So this is some work done at Dalhousie University by Camilla Mora. And so here's a discovery curve for fishes. And what he's shown here, this is the rate of new discovery. And then he's used a variety of statistical extrapolation tools to carry that line out further. Now, the different models don't particularly matter. If any of you have done chemistry, you may scream when you see Michaelis-Menten, but nonetheless, they're extrapolation methods based on the shape of the curve. And so, based on this, they came to a relatively satisfying conclusion that we know about 16,500 species of marine fishes, and there are somewhere between 1,000 and 4,000 more to go. And so we still have maybe 25%, maybe as little as 10% of fishes that remain undescribed, but we're getting there. You know, we've made some pretty good headway. And he's even told us where to look. He said, look in the tropics, look in the deep sea, and you'll probably find many new species in those environments. Now, if we look at these threadworms, these little worms that I told you should know about, but you may not, it's a little different story. So these are deep sea nematodes, and here we see a discovery curve that some deep sea biologists put together. And if we try and fit a curve to it, we could say, well, maybe it'll look like that. But equally likely, we could say it'll look like that, or equally likely, we could say it'll look like this, and again, shoot through the roof of the building. So we simply can't say based on this technique. Now, it turns out there are other ways to do this. They all have flaws to try and extrapolate. One of the best ways is to talk to taxonomists and say, what do you think? Based on what you've seen when you go into these new environments, what proportion is new? And so what they say is there's probably about 100,000 more to go. Now, because I'll probably forget to say it later on, we know of about, I don't know, 500,000 species or so. For the oceans, we're moving towards about 500,000 species of described known species. But in fact, I think the numbers that actually have names on them are much smaller. But we think the number will go above 1 million. So most of us think that at best we're halfway, and that does not include the microbes. The microbes are a real wild card, and I'll show you why in just a moment. So it turns out that we know a fair bit about big things and not a whole lot about small things. And so this is a graph that shows you the unknown is this area of white, and what we know about is in blue or in orange. And so of the things we know about, some of them are things that we actually do monitor. So for example, we try and keep track of fish populations. Down here, these are E. coli and the phytoplankton that cause harmful algal blooms. So those are things that we monitor for, which you see are shown in orange. But the ones we actually try to manage is even smaller in terms of its proportion. So if we look at the global diversity on Earth, you should think of this broad area here, the global diversity in the ocean. And this is the part that we actually try and pay attention to. So there's a lot that we ignore. So how do we do better? Well, in fact, the census worked to develop a lot of new tools to try and do better with this diversity problem. The first of these was something called the Ocean Biogeographic Information System. And this is a data set, a database, that pulled together a lot of the data from the census. It pulled together a lot of other data sets on biodiversity in the ocean. And so what you see here is a map of the number of records for different areas around the world. So through a lot of hard work, this group has now assembled something like 30 million plus records of life in the ocean. Now by a record, what I mean is if we know that a certain species was collected in Boston Harbor, then there'd be a point in Boston Harbor for that species on this map. And so 30 million records, and what this has shown us is some really key geographic gaps. So not surprisingly, the areas in yellow and red are the best sampled areas. So Northern Europe, the east coast of North America are pretty well sampled compared to the rest of the ocean. But vast areas you see that are either blue or black, meaning we have nothing from some of these locations. This is a cutaway view of the ocean. You can imagine it. So this would be the shoreline, this would be the deep ocean, and this is the area in between. So this is the surface layer up here. And so what you see is this blue area is, again, where we know almost nothing. So the deep layers of the ocean, lots and lots of new potential there. One of the things that I often say when I give a version of this talk is that if you want to collect new species, it's easy. You can just go out into the deep ocean, drop a sampler down, and bring it up, and it's very likely there'll be something in that that's new to science. And so that's not the challenge. Though certainly there are challenges associated with that. So there are big geographic gaps in coral reefs, which are, we think, very diverse, but very undersampled, as I illustrated with my Nancy Knowlton example. The deep sea midwater layers, so not quite at the bottom, but just above it. And also the seafloor, so these deep sea layers down here, again, very poorly sampled. And then finally the poles. One of the interesting things about the poles, and the Arctic in particular, is that this is an environment that's been covered by ice for many, many years. And so humans have not been able to sample or fish or drill for oil in many of these areas historically, and it's about to happen. And so some of the most pristine habitats on Earth are about to become accessible. And so we are hoping that people will work very hard in the coming years to make sure we get some baseline data. As I told you about before, we typically do not have baseline data for most of the ocean because human exploitation has been going on for so long. Our knowledge is quite uneven, and so here we see an example of coral reefs. The estimates on coral reef diversity is something between 0.6 and 9 million species. Again, people debate these numbers, but the point is, very small area of the Earth's surface covered by corals, but perhaps the single most endangered environment in the oceans. And so there are concerns that within the next 10 to 50 years, we could see major changes in coral reef ecosystems because they're very sensitive to temperature, which of course is increasing, and they're also very sensitive to ocean acidification. And so lots of big concerns and urgency associated with coral reefs. Now, one of the other tools we now have available to us is something called barcoding. And if you've ever been to the grocery store or even a hardware store, and you see the barcode on an item, and you can go and scan it, and it'll tell you what it is and how much it costs. Likewise, molecular biologists have developed something called a CO1 barcode, and essentially it's a unique barcode. It works for most species, not all. It doesn't work for microbes, for example. But what this means is that each species has a unique identifier, a unique barcode. And so what this means is that if you have this pelagic snail that you collect in one area of the ocean, it will have the same barcode no matter where you collect it. Now, what I found is one of the most interesting applications of this technology was actually a project by two young ladies working in a high school project. They worked with the census, and they went out to fish markets and restaurants in New York City and collected fish. And they brought those fish back and they barcoded them. And they found all sorts of interesting numbers. So for example, they found that white tuna, which is worth about $8.50 a pound, it's probably gone up by now, was being sold in some cases as tilapia. I mean, vice versa. So tilapia is being sold as albacore tuna. And so you're paying $8.50 for what you think is an expensive fish, and in fact you're getting something not worth nearly as much. They even found an endangered species being sold in one of these fish markets. Again, it was labeled as something else. But the point is, there is a lot of this sort of stuff going on. And talking to the leaders of the Barcode project, I was having lunch with one of them a couple of months ago, and he said, you know, he works in Canada as well. And he said, you know, if you go to seafood restaurants and regularly order seafood, I can guarantee you that you will be eating stuff that is not what is labeled. Not always, but if you do it on a regular basis, there will be substitutions, there will be mistakes. Some may be deliberate, who knows what goes on. But the point is, barcoding allows you to get around that sort of problem. You can know exactly what you're eating. He actually said, too, that if you, even if the fish is cooked, you can still barcode it. So you can take that fish home and get a barcode and find out if you got what you paid for. The reason this is so important is that we have a lot of species that are very difficult to tell apart. So blue mussels, you've probably all seen blue mussels along the seashore, are in fact several species that look very similar. So there's Middleus edulus, which is a very common species, but there are two other species, Gala provincialis and another one that look very much the same. And based just on the shape, the morphology, and measurements, most people can't tell them apart. But if you use something like barcoding, you can tell unambiguously who's who. And so that can become very important if you're trying to do studies of the geography of these species. These guys were actually used to monitor pollution in something called a mussel watch program. Well, if you're confusing your species, they have different metabolisms, different growth rates, and so they're going to give you very different numbers if you're looking at two different species. You need to know that if you're using them to monitor pollution. So that's barcoding. There is a variation on the barcoding concept called 454 pyrosequencing, and this is for the microbes. Now, the microbes mostly look like this under a microscope. Not very charismatic, not a whole lot of features to try and tell them apart, although there are some that are actually quite distinct. But the point is, historically, and certainly the training I received in going through school, is that microbes are abundant, they're hugely important, they're everywhere, but they're not very diverse. And the reason for that is, back in the bad old days when I did my schooling, we only really had morphology and also growth. So if you could grow these microbes on agar or another surface, and then look at the characteristics of the growth rates and the shapes of the colonies, that gave you different means of differentiating between them. But now we have a genetic tool called 454 pyrosequencing, and that tells us which species is which. So based on this, we're suddenly recognizing a huge diversity, up to 1 billion different types of microbes in the oceans. So this is a fundamental change in our way of thinking about microbial diversity in the oceans. And so this allows us to start thinking about biogeographic studies, evolutionary relationships among groups, but we're really literally just scratching the surface. We've just realized this in, I would say, the last 5 years even. This has become apparent. What else can one do with this sort of data? Well, this is some work again done out of Dalhousie University. Derek Tittensor is a postdoc. So he took some of the data from the Ocean Biogeographic Information System that I showed you earlier, and he asked the question, where are the hotspots of diversity? So if we are going to prioritize some areas of the global ocean for conservation, where ought they to be? And so when you look at coastal taxa, and this is for things we know relatively well, so relatively large things, fishes, marine mammals, some crustaceans and other groups. But what we find is that for coastal taxa, and this is no surprise, we see this huge diversity in the tropical Pacific in something called the Coral Triangle. This is an area we know is extremely rich in species, so that wasn't a real shock. But what was surprising is for the oceanic taxa, we actually see peaks that are away from the tropics. And so often temperate sort of latitudes like New England can have the highest diversity of some of the groups of organisms. So that tells us, depending on what we want to conserve, we may need to think about different sorts of strategies. And one can do this on different scales. So this is a global scale. But you could do a similar sort of thing, for example, on Georgia's Bank, if you have sufficient data, or in the Gulf of Maine, to predict which areas are likely to be hotspots for biological diversity. Now, one of the other classic examples that I like to put out there is this one. And so, one of the early criticisms of the Census of Marine Life is that it was a stamp collecting exercise. So you've got lots of diversity out there. Who cares? You know, we only eat so many species of fish. The rest of it, why do we care? There was a scientist working at the Hart Institute in Texas named Wes Tunnell, and he was very interested in the Gulf of Mexico, and he decided as his sort of last major undertaking as a scientist would be to bring together all of the known data on the Gulf of Mexico. And so he created this huge tome. The book is about this thick, I kid you not, all the information he was able to pull together on the Gulf of Mexico. And at the time, it seemed like one of the most esoteric exercises in the census. You know, why would you spend all this time working on this project? You know, it's a great database for scientists, and we were very excited about it, but it seemed very esoteric. Then, of course, we had the Deep Horizon oil spill. And so all of a sudden, this data that he pulled together, just out of interest, out of scientific curiosity, is very likely to become one of the single most important resources in the lawsuits that are going to go on for the next 20 or 30 years, multi-billion-dollar lawsuits over this oil spill, and he's got this data set that was collected really for fun. And so it illustrates the role of baseline data, how important it can become because things change. We run into things we can't predict, and so if we have the information, we can start to know what damage has happened and how quickly it's going to recover from that damage. Now, this is one of the harder concepts to try and relay, so I'll do what I can with it. And so one of the other ways to look at biological diversity is to ask the question, does it matter that we have a bunch of species? So again, we only eat so many of them. What do these other species do out there? One of the things that we started to focus on as ecologists is something called ecosystem services. And these are the things that the environment does for us at no cost. So it produces fish, and we don't have to throw anything in, it just produces fish. It cycles nutrients that drive the whole food chain. It breaks down pollutants, including the oil. There are microbes that break down oil. And so all this stuff is free. And so the question is, what is the role that biological diversity plays in these services? So this is a study done actually by Italian scientists that looks at the diversity of different types of animals and the way that they feed and how that affects microbial production. The long and the short of it is that the more diverse animals you have, the higher the microbial production. Why is that important? It means the nutrients are being cycled at a faster rate, carbon is being broken down more quickly, and you asked that question early on about different areas of the oceans and their productivity, comes back to the regeneration of those nutrients. And so we have all of these cycles that if you break the cycles, the oceans get into trouble. As long as the cycles function, you're in great shape. So here we say, it looks like diversity plays a role in keeping these cycles moving. And if that's true, then we should really care about diversity, even if we don't eat any of these guys. So the long and the short, more species means more production. That is good for the things that we extract from the oceans. One of the other sets of tools that was used widely throughout the census was acoustic sound waves. We can use sound waves for a whole variety of things. Now, one of the things I mentioned at the onset is that water, though we think of it as clear, is not. Light does not penetrate very well through water. Sound waves do. They can actually penetrate quite well. And so the census actually used sound waves for a variety of different applications. Now, one of these is to map habitats. And so this is something called multi-beam imagery. And the idea here is you bounce sound waves off the seafloor, they bounce back up, you receive them, and based on the characteristics of the bottom, it gives you a different sort of signal. And what you can do from that is to create these very detailed maps. Here you see the Bay of Fundy and the Gulf of Maine. This is actually a bay on the south coast of Newfoundland. And so you can start to create very detailed habitat maps. Why does that matter? Because, again, something that I would say is unknowable is the distribution of all species. We will never know where all species live in the oceans. But we can know where all the habitats are. And if we know that certain types of species like particular habitats, we can start to predict where we're likely to find them. And so this sort of thing we can do relatively quickly. This sort of map one can do in a period of weeks to a month. If you were to try and sample all the biology in that environment, it would take you many, many years. So we're looking for shortcuts. And so we can use acoustics to do habitat mapping. One of the other tools, actually this is work done up at MIT, Nick McCrease, just up the road. And so this is using sound waves to count fish. Now in the bad old days, the way we would count fish is to put a net over the side, do a standardized tow, and see how much fish we caught. And based on that, we would estimate how many fish are in the ocean. There are all sorts of problems with that, not the least of which is you have to kill a lot of fish in order to get your estimate. Now, what we can do is to use sound waves. And so what McCrease did, and this actually is fundamentally a leap forward. We've used acoustics in the past, but this is really quite novel because of the scale at which you can do it. So you have one ship that's sending out low-frequency sound waves. You can see here we're sending out sound waves. It bounces off the fish and then comes back to the receiver. The receiver can then actually tell you how many fish are there. So this is an area about the size of Manhattan Island. And these are large numbers of what we think are very likely herring that he was able to image in a period of about a minute. And so being able to image an area of Manhattan Island in about a minute and to get an accurate count of the numbers of fish can really revolutionize fisheries management, because one of the key unknowns and one of the key problems we have in managing fish is to know how many are left in the sea. And so if we take too many, they start to decline. And so if we know how many are there more accurately, then we can fish with greater confidence that we're not overdoing it. Again, coming back to a very interesting fisheries application, this is work done out of Vancouver Aquarium on the west coast of Canada. And what they did is to put out a series of listening curtains. And so these are hydrophones that listen for these little radio tags and other types of tags. They're about the size of a pencil eraser, and you surgically implant them into fish, and then put the fish back into the wild. The reason this is very important is because it allows us to know what the fish are doing in the ocean. Now for salmon, we know a lot, but almost everything we know about salmon is from fresh water. As soon as they hit the ocean, it's a black box. We know almost nothing. So what these guys have done is to set up these listening curtains at key bottleneck areas around Vancouver Island, and so they can actually see when those fish pass by. Because these little radio tags are unique. Each fish has its own special radio tag, and you can say, ah yes, that's the fish that I set loose in Seattle back in October of last year, as opposed to this fish which I released from Victoria. And so it allows us to get very precise estimates of where the fish are moving and where they're dying, which is something we did not know before. As a side story to this project, they also tagged some endangered green sturgeon, and they found that these green sturgeon move back and forth along the west coast of the US and Canada. And so again, if we're trying to manage this species, knowing where it moves is really just the starting point of the information that we need. And so these are really very fundamental new breakthroughs in the way that we can follow life in the ocean. And again, acoustics is a very key part of this. Now, one of the other things is what we've been calling blue highways, hotspots, cold spots, and truck stops. And the idea here is you see this leatherback turtle, and it has on its back this little device, which is in fact a little GPS with sometimes some sensors on board. It also has this little antenna, and what that is going to do is it's essentially a cell phone that's going to call home and tell you where that turtle is. And so what happens then is as these animals swim across the ocean and they come up to the surface to breathe, they exchange information with satellites, and we can track them right across the ocean. And so this Tagging of Pacific Predators project tracked a whole bunch of different species. You can see the list down here. There's more than this now, but it illustrates the point. And you can see these key areas of the oceans where lots and lots of species move. Now again, that can tell us where we need to be careful in terms of fishing gear if we want to protect species such as leatherback turtles. We can also use these animals as oceanographic ships. And some of you may have followed Stelliff Colbert. If you've ever watched the Colbert Report, it's a television show. And they actually named an elephant seal after Colbert and monitored it in real time. So every day you could check in and see where Stelliff was living in the ocean. And so Colbert himself really latched onto this and started broadcasting it as part of the show. And it was the greatest publicity that they could have hoped for, but it was very successful. And so what happens then is if the the tracker that you have on the animal also has sensors, in this case temperature and salinity. You can start to collect oceanographic data from remote parts of the ocean, and the animal does the work for you. So to send a ship out to this part of the ocean would be hugely expensive, millions of dollars. But if you attach it to one of these elephant seals and they swim out, and then they come up, they surface, they send their data back via satellite, all of a sudden you have a very cheap oceanographic ship. And so this is in fact one of the new tools that's become very intriguing, very widely used, and potentially really groundbreaking. So then we come to predictions past and future, and this is actually the project, well it's actually two projects, but one of the things I found most intriguing about the census. And so the scientists here wanted to know what the oceans looked like before scientific data. And scientific data really began, for the most part, around World War II. There's not much that goes further back than that. And a lot of it is even more recent, sometimes the 1970s. And so they used anything they could get their hands on. They used things like monastery records, restaurant menus, ship's logs. All of these things often referred to whatever species were on the go, what they were catching, what they were consuming, and from that they tried to piece together what the oceans used to look like. This is actually a really nice piece of work. A graduate student, Lauren McClenahan at Scripps Institution, looked at trophy fishes from the 1950s onward. And so here you see 1950s, these pretty impressive looking fish that were winning the prizes at that time. And then we get to the 1980s, not quite as impressive, but nonetheless, these are the trophy winners that we see at that time. And then we get to 2007, and yeah, I mean, it would be laughable if it weren't so pitiful that these are actually the trophy-winning fish. And you know, if you had brought these fish back in the 1950s, you would have been laughed at because you had such a pathetic catch. And yet that's what it's come to. And that's really just in a period of 60 years. The reconstruction of what the oceans look like revealed some very interesting things, one of which is the role of the Romans. And so the early Romans, some of you know something about Roman history, but in the bad old days, fishermen would go out and they would catch their fish, they would bring them back to shore, and they would either eat them or sell them right away. That's all they could do. There was no refrigeration, there's nothing else to be done with those fish. Now the Romans were a clever lot, they came up with salting. And so once you discovered a means of salting fish, that meant you could preserve them. So once you preserve them, you can start to transport them large distances, you can store them for many months before you put them on the market, and so industrial fishing begins. And so again, although we tend to think of the 1950s maybe as the start of industrial fishing, the Romans were actually at it back in Roman times. And so depending on where you are, human impacts were very substantial in many cases, thousands of years ago, not tens of years, but thousands of years. Some of the other sorts of sources that we can use, this is a painting by Franz Snyder, a Belgian painter, and this is a fish market, and you can see all this variety of ocean life. And he was known for realistic painting, he didn't do surreal. And so we think this is a relatively realistic depiction, and yet a lot of these things would not be found in the North Sea today. And yet look at this diversity of life that was on the fish market at that time. And then of course the example I gave you earlier of this Danish fish market, look at all these tuna fish. Now, one other little statistic I wanted to mention to you, some of you may have caught this on CNN or another news channel. It was about 3 months ago. A tuna fish sold a single tuna fish sold at auction for $370,000. That's for one fish. Now look at that sort of abundance. And again, the fact you will not find a tuna in the North Sea very easily these days. So it raises 2 points. First of all, one is the depletion, but second of all is the demand on these resources. You can imagine people are going to be willing to kill for $370,000 payoff, you know. And yet these fishes move between international boundaries. Well, they don't care where the Canada-U.S. border is or where the Portuguese fishermen are likely to catch their fish. They are just moving. So this is some work by Heike Lotze and Boris Worm at Dalhousie University looking at trying to look at depletion as a function of time. And they used a whole variety of sources. And the long and the short of it is that we see declines anywhere from 65 to 98% in these major groups of organisms from historical levels. And so that includes everything from fishes to seals and whales to sea turtles to coastal birds. Now the gray areas are actually a cause of hope. The gray areas are recoveries. And so if you see some of these, they're all blue, so it's all bad news. So they decline by 90%, they're not recovering. In the case of some of the marine mammals, which we have chosen as a society to try and protect, we do see evidence of some recovery. It's the same with coastal birds. So there is some hope, if we can keep our mitts off these things, that they can recover in some cases. Now, I will say that in the case of the Newfoundland cod, we have mostly kept our mitts off them, and 30 years later, there's still no recovery in sight. That fishery remains largely closed. One of the other examples, and I'll just go through this very quickly, there's a lot of detail on this, but this is the abundance of great sharks in the Gulf of Mexico. And all you need to get from this is down, down, down. So the horizontal axis is time. And so the time period is the 1970s through 1990. Everything is in decline. So these large great sharks are being fished down. No great surprise there. What's kind of interesting in this example is that we have small skates and rays increasing in abundance. And the reason for this is we've plucked out their predators. And so because we've allowed these mesopredators, as we call them, to increase, they in turn feed on scallop. So what we've seen is a decline in scallop as a result of overfishing great sharks. And so it's not very intuitive. I mean, what we've done is one type of fishery, which we think is completely unrelated to another, is in fact causing major declines. And this is what we call a cascading effect, where something cascades right through the food web. So perhaps the most significant tool in many ways that the census produced is a united and enthused scientific community. I mentioned 2,700 scientists from around the world working together. There's a core group of, I would say, 400 or 500 who are very active in the census. And this group also managed to gather the interest of policymakers and also intrigue the public. Some of you have probably seen the Yeti crab. It was featured in a lot of magazine articles. The name is kind of obvious with all of the hair. This was found near Easter Island, near a hydrothermal vent. Spectacular organism, and it captured so much imagination. There are stuffed Yetis that you can buy out there. There's Yeti artwork, all inspired by this critter. And this comes to the point of communication. And now, New England Aquarium is very different from my sort of environment, which is a university. And university scientists are really taught this sort of thing. I'll read it for you. One thing I'll say for us, Meyer, we never stooped to popularizing science. And that is the way most university scientists think. It's the way we're trained. We should publish papers, and that is it. Places like New England Aquarium, very different view, obviously, trying to make ocean awareness increase to whatever extent possible. The census took the view similar to the New England Aquarium, that the public ought to know. And so a whole variety of outputs from the census tried to communicate the findings of the census program to a whole variety of audiences. And so there are the Oceans film. Some of you may have seen this. There's a Disney version, which is sort of a not-so-great documentary, but the European version is really wonderful if you ever get a chance to see it. And in both the Disney and the European versions, the imagery is just spectacular, beyond words. A variety of books, some for a scientific audience, some of these for a more general audience. This is actually Sherman's Lagoon. Some of you may read this comic strip. They did a whole series on the Census of Marine Life. Jim Toomey did that for us. And there are a whole variety of newspaper and magazine articles about the census, including there's the one about the derby fish. So the census invested a lot in communication, and it was very successful. People have heard about the census, they've seen a lot about marine biodiversity, and they're starting to care. So what about the future oceans? Well, I'm not clairvoyant, and I certainly would not predict the end of the world and fail, as recently happened. But I am willing to predict what I think things will look like in 2020. That's not that far away. So first of all, I think it's going to be more crowded. We're going to be extracting more energy. We'll be transporting more goods across the oceans. There will be more people living along the coastline. And as our thirst for internet increases, more and more communication cables across the seafloor to make that happen. These will all contribute to more environmental concerns. Global warming will become an increasingly worrisome issue, along with the other impacts that I described earlier. And these will lead to more rapid biological changes, including species loss, habitat loss, and a whole variety of other concerns. There are also likely to be more ocean conflicts. I mentioned this $370,000 tuna. People will be fighting over ocean resources. And even among friendly nations. I mean, right now in the Arctic, Canada, the US, Denmark, and Russia are all squabbling about where the international boundary ought to be. And it's primarily driven by seabed resources. There's oil in them by our hills, and everybody wants it. There are, in many instances, good endings. Canada and the US had a long-standing conflict over George's Bank, which was resolved without us going to war. But nonetheless, the pressures are going to increase. But there is one bright spot, which is that we will have more information. So all of these tools that I've been talking about will make the oceans more transparent, and it at least gives us the potential for better planning. Whether we use this potential is up to us, it's up to our lawmakers, but nonetheless the information is there, and we can do better if we choose to use it wisely. So pressure on the ocean will worsen, it will not lessen. And so I would argue that what we need to do is to know what's there and how it's changing. Census has helped a lot with that, but there's still work to do. We need to figure out which species really make the oceans work. This issue I brought up with ecosystem services is a brand new science. That's less than a decade old that scientists have really worked on this particular question. And then finally, we need to provide managers with scientific strategies for sustainable oceans. If we know we're going to continue to use them, and realistically we will, no matter how conservation-oriented we all are, we know that we can't stop using the oceans. There are too many of us. And so the question then is, how can we use them more sustainably? And so if we can provide managers with better information for better strategies to manage those oceans, then perhaps we can sustain them more clearly. So this talk is really focused on the Census of Marine Life, but one of the questions I've been asked the most as the census came to an end is, what's next? And the answer that I've had is that there are projects that will continue on, smaller projects on a very small scale. Nagiso, for example, some of the work that they've done with the Newfoundland Aquarium will continue on at a small scale. But as a global endeavor, the census is over in its current form. There is a program in Canada that we've started up. We are hoping to see similar programs develop elsewhere in the world. This is Canada, the coastline you see here. We are a very interesting nation because most of our people live right here, jammed up against the US border. The only large Canadian city that's on the ocean is Vancouver, and so that's the only voting power we have. Most of Canada is in central. Our national capital, Ottawa, is landlocked, so we have to convince the people of Canada that they should care about the oceans. It's a harder struggle because our population is so differently distributed than the US population. And we have fewer people. We have about a tenth of the US population and tight budgets as we have in the US. And so we've created this national network called the Canadian Healthy Oceans Network that's trying to carry forward some of these objectives. And I'm not going to go into any detail on them. But the point is that the tagline for the program is biodiversity science for sustainable oceans. And so what we're doing is we're taking discovery-based science, just like the census has promoted so effectively for the last decade, and saying, what can we do with it next? What can we do with that information to make oceans more sustainable? And so there's a group of us from the census who've been tasked with trying to create something, try and generate something new that will take advantage of this huge science community that's working so well together. They're mobilized. They've already got the tools, so a lot of the footwork's been done. If we want to try and find ways to take that community and apply the knowledge to real problems. And so we've come up with a program we're calling Life in a Changing Ocean. And the scientists are the Global Marine Biodiversity Consortium, which will be an international group of scientists committed to expanding marine biodiversity knowledge to support healthy and sustainable oceans. And so we are just starting to form our plan. We've got 4 major research themes, which I'll put up here. Ecosystem services rears its head once again. Ocean spatial planning, where do you put your MPA so it's most effective? That sounds like an easy question. It's actually a very hard question, and often not driven by science. More often driven by other factors, economics and ease. Biodiversity observation. We know the oceans are changing. We need to be able to observe the oceans better than we have in the past. Satellites tell us very quickly if land use is changing. We don't have a similar sort of thing, particularly below the surface layers of the ocean. And then finally, how can we ensure biodiversity is maintained given the many pressures that we place on the oceans? And how can we ensure sustainability? So that's what Life in a Changing Ocean, if it ever takes place, we're just starting to try and launch it, that's what it will try to address. So I've been talking for quite a while. I'm going to shut up now, but I'd be happy to answer any questions, and I thank you again for coming out on this.